So I left off, we were talking about species and speciation. And I talked about, first of all, Meyer's biological species concept, which defines species as entities, sets of populations that can all potentially interbreed and produce fertile, viable offspring. So the usual example is horses and donkeys are separate species because their offspring, mules, are perfectly fine living things, but they're sterile. So horses and donkeys cannot permanently exchange genes. We say that they're reproductively isolated. Meyer also mentioned a mechanism for speciation that he proposed, and the model is called allopatric speciation. And you start with a population of one species that gets split by a physical barrier that individuals can't easily cross. They might occasionally manage it, but they cannot routinely get across the barrier. One possibility is that a few organisms from one population migrate across a barrier that they don't normally cross and set up a new population on the other side of the barrier. That's called dispersal. So if you have a species of bird on one island and a few of those birds get blown across the ocean to another island, that's too far away for them normally to make the crossing, that would be dispersal. If the organisms stay right where they are and the barrier comes up within the range of the population, that's called vicariance. For a while, there was the evolution prof at Hendrix studied these mountain dwelling, they're not rodents, they're actually related to rabbits, but they've got short ears. Animals called pikas in the Western United States, Pika, pikas. And back at a time when the climate was cooler, pikas were able to live in mountain ranges and also survive in the valleys between the mountain ranges. What has happened in the past 10,000 years is the climate's gotten warmer. The valleys between mountain ranges are now too hot. The pika populations are still living at high altitude on the mountains. The pikas haven't really moved, but now you have these separate pika populations that can't cross these valleys. They used to be able to when the climate was cooler. Now they can't, and you have isolated pika populations that are stuck on these mountain ranges. The barrier in this case being this zone of hotter climate that's cut them off from each other thanks to climate warming over the past 10,000 years. That would be vicariance. Heck, found a paper somewhere showing there was genetic differences in snake populations now on either side of interstate highways. What's happened is that the snakes, haven't particularly moved. What's happened is that people built an interstate highway through the range of these snakes, and now snakes on one side of the highway cannot easily get across to mate with snakes on the opposite side of the highway because they get squashed. So that would be another case of vicariance. If the organisms don't move and the barrier comes up and divides them. Now, those separated populations will tend to become different. Natural selection may shape them in different ways. Alleles in one population can't be exchanged with the other population. Mutations in one population can't make it into the other because gene flow is blocked. If both populations are small, genetic drift is going to change allele frequencies. And one population may have an unusually high or low frequency of certain alleles purely by dumb luck. That's called the founder effect. So these populations are going to tend to become genetically different from each other. And given enough time, the separated population should diverge into separate species. And even if that barrier disappears, they should not be able to interbreed. And then we truly have separate species. Here's the thing. We talked about different isolation mechanisms that keep two populations from interbreeding. And the problem, not the problem for life, but the problem for our understanding of life, is that these isolation mechanisms aren't absolute. And I gave you the example of crosses that have been made between domestic cats up at the top and servals, a small wildcat native to Africa. That's a serval down there at the bottom. At least as of a few years ago, there were some servals in the Little Rock Zoo. If you ever want to see some relatively up close and personal and you can't make it to Africa. And when this was tried, there were a number of isolation mechanisms. Servals often didn't want to mate with domestic cats. Maybe they smelled funny or something like that, but servals weren't too keen on the idea. The gestation periods are different, and that can cause pregnancy problems. And hybrid litters were often miscarried, absorbed into the mother's body, which humans don't do, but some animals can, or born prematurely. And yet, it worked often enough that F1 hybrids have been produced between domestic cats and servals. Now, F1 hybrids are still not very good pets. Their behavior patterns are still too wild. But the F1s were back-crossed to domestic cats, and now there's a population of basically F3 and F4s that may only have roughly an eighth or a 16th of their DNA coming from servals. And these are cats that have the body shape of domestic cats. They are easy to take care of, like domestic cats. Well, I don't know if domestic cats are particularly easy. I've got rather strong opinions on the matter. But at least they don't completely shred your house the way that a serval in your home would. So they behave like domestic cats. They just have the wild-looking coat patterns that people find very beautiful. They look like wild animals, but they're not nearly as difficult to care for or as, ethically problematic as caring for a real wild animal would be. But it raises the biology question, have we got one species or have we got two? I mean, they don't normally interbreed, domestic cats and servals, but they can. And much of the time it doesn't work because there's isolation mechanisms in the way, but occasionally it does. Incidentally, this has been done a second time. This is a Bengal cat, and this was created in much the same way. Domestic cats were hybridized with a small cat species called the Asian leopard cat, Prionolurus bengalensis. And then the hybrids have been back-crossed to domestic cats for several generations, giving you, again, a cat that's well enough behaved that you can have it in your home, but that has, this beautiful, wild-looking coat pattern to it. So beautiful, wild-looking cats, but they're not nearly as hard to care for as real wild cats, and they're also not endangered or otherwise, ethically questionable. So no problems with Carol Baskin. When this happens, we call this introgression. When you have some hybrids that are back-crossed with one of the parent species. So when the F1 serval domestic cat hybrids are mated to domestic cats, and the resulting F2s are mated to domestic cats, and so on. So we have some serval DNA that's kind of trickled its way into the domestic cat population. And savanna cats with serval DNA are pretty expensive. But if it hasn't happened already, I'm sure it's just a matter of time before a male savanna cat runs out and knocks up a bunch of female cats before they can get him neutered. And we'll eventually have just a little bit of serval DNA kind of trickling its way into the feral cat population as well. And again, that's called introgression. Just a, yeah, rare hybridization events allow DNA from one species to trickle into the gene pool of another. And this can be important in wild species, and it can be an important source of genetic variation. This is a way that species can sometimes pick up new alleles, is by rare hybridizing with other species and then introgression. So here's an example. We generally consider brown bears and polar bears to be different species. They look different. They've got very different niches. They've got very different diets. Brown bears will eat a lot of berries and insects and things like that. Polar bears are specialized just about exclusively for large mammals, like seals and sea lions and Canadians. They look like they ought to be separate species. In zoos, you can hybridize them. It has happened. I don't think they do this deliberately, but there have been cases of it where polar bears and brown bears will hybridize and produce fertile offspring. And this is really weird. When they started doing genetic studies of wild brown bears, in particular, brown bears from islands off the southeast coast of Alaska. For short, they're called the ABC Islands, and I think that might stand for Aleutian, Baranov, Komandorsky. Anyway, it's 3 groups of islands that happen to have names beginning with A, B, and C. So when they looked at brown bears from these 3 island groups, they turned out to have DNA that was more similar to polar bears than it was similar to other brown bears. So there's evidently been quite a lot of hybridization to give you populations that look like brown bears but have an awful lot of polar bear DNA. The most recent finding is that polar and brown bears have mostly, not exclusively, but mostly distinct nuclear genes. And we estimate that the split between brown bears and polar bears would have happened about 600,000 years ago. Incidentally, that's well before humans were anywhere near them. That's before the origin of modern Homo sapiens, I might add. The problem is if you do a cladistic analysis, like we did with the seashells, but you do it with mitochondrial DNA, polar bear mitochondrial DNA is nested within the DNA of brown bears. Now, think about this. Mitochondrial DNA is passed on only from mother to offspring. It's only passed on in the female line. So the mitochondrial DNA of all polar bears is nested within that of brown bears. That means that the mothers of polar bears, on the face of it, it looks like she was a brown bear. That's a little weird, but further analysis, it turned out that what happens is that brown bear males have been mating with polar bear females for thousands of years. And this happens because brown bear females tend to stay in the territory where they're born. Males tend to get out and roam. So brown bear males are the most likely to encounter female polar bears. So brown bear males have been mating with polar bear females, and that's created hybrid populations that look like brown bears. They mostly have brown bear nuclear DNA, but they've got female ancestors that were polar bears. There, in some of these brown bear populations, up to 9% of the nuclear genome originated in polar bears. So this is kind of weird. Let me do this one more time. Polar bears and brown bears split, but in the centuries after the split, male brown bears have mated with polar bear females, and that's produced a population of stable hybrids that have mostly brown bear nuclear DNA, but they still have mitochondrial DNA that's typical of polar bears. They have female polar bear ancestors, if you go back far enough. And incidentally, there's no evidence for hybridization going the other way. There don't seem to be cases of male polar bears mating with female brown bears, possibly because male polar bears don't roam in the same way that male brown bears do. And that creates an odd problem for the biological species concept. What do you do when you have separate groups, separate populations that can exchange genes, but they're not doing it symmetrically, where males of one mate with females of the other, but it's not the other way around? What do you do when, as is often the case, not all hybrids are sterile? When you have, let's say, sterile males but fertile females, as sometimes happens. Yet another problem with the biological species concept, stepping away from bears for a minute, is that the biological species concept is not easily applicable to organisms that don't have sexual reproduction at all. Most amoebas, that's an amoeba, that's a big eukaryotic cell, single cell protist. Most amoebas have never been observed reproducing sexually. If they're doing it, they're extremely discreet about it, but we've never caught them at it. There's a whole phylum of animals called the rotifers, and one major subgroup of rotifers, the bdelloids, for at least the past, well, the fossil evidence says 25 million years, and I think there's molecular data that pushes it back to something like 80 million years. But for millions of years, they have not reproduced sexually at all. They're all female, and they reproduce by parthenogenesis, which is a way of creating a female can create an egg that's already fertilized and ready to divide. There's various tricks that females can use to do that. So what do you do with about 400 species of bdelloid rotifer that have been reproducing without sex for 80 million years? And what do you do with bacteria? Bacteria do reproduce sexually. That's two E. coli in the picture down below going at it. One of them has grown a little tube called a pilus and is sending genetic material through that pilus to the other cell. So, sex is basically mingling your DNA with somebody else's and seeing what happens. And that's what these bacteria here are doing. But bacteria can incorporate DNA from almost any source. If you remember Griffith's experiment on the transforming principle, it was a critical piece of evidence that heredity is the result of something physical, something that turned out to be DNA. That experiment revolved around living bacteria picking up DNA from dead bacteria. Bacteria can pick up DNA from anywhere. Bacteria can swap pieces of DNA around like they were Pokemon cards, or, I don't know, Dragon Ball Z cards, if that's your generation. A microbiologist named Sorin Soneo once observed that if you strictly followed the biological species concept, there was only one species of bacterium. That's not particularly useful, to diagnose and treat disease, for example. You have to keep track of dozens of different species of bacteria. but it doesn't look like you can really use the biological species concept to define those species. And then sometimes the following the biological species concept where it leads gives you answers that look funny. On the left is the eastern blue jay, Cyanopsida cristata. This is the one that you've seen, all your life if you grew up in Arkansas. We have these birds; they're very common around here. On the right is that's a male Steller's jay, Cyanopsida stellari, a bird that I used to see when I was in grad school in California. That's a Western species. Now they look different. You can easily tell them apart. They live in different areas, except they overlap in a small part of Colorado. And where they overlap, they do produce some fertile hybrids. There is what we call a little hybrid zone where the two species meet. So do we have one species or two? Gene flow between these two is restricted, but it's not absolutely blocked. One moment. I love you, darling. Have a good day. I'll see you soon. Right. On the flip side, we've got cryptic species like western and eastern meadowlarks, which you could see in Arkansas. Physically, they are virtually identical. There's no consistent features that enable you to tell them apart, but they don't interbreed, and they're separated by the fact that they've got very different songs. That's probably the main isolation mechanism between them. Males of one sing a song that females of the other just simply don't recognize. And when you've got two species that are reproductively isolated from each other but don't look different, we call those cryptic species. So western and eastern meadowlarks are an example. If you know your birds, Carolina chickadees, which are what we get around here, and black-capped chickadees, which we get up to the north, are another case of cryptic species. They look virtually identical. You can't consistently tell them apart, but they don't interbreed. And then you get things like a small African antelope named Kirk's dik-dik. Instead of one species, you've got three populations that are different because they've got different karyotypes, different chromosome numbers. So these three differ because they've got chromosome fusions, inversions, translocations. That's when a piece of one chromosome ends up stuck onto a separate chromosome, and so on. And if you cross individuals with different karyotypes, you get sterile hybrids, even though so we've clearly got three species here by the biological species concept, even though the two of them look, even though they all look completely identical. You can't tell apart the three karyotypes of Kirk's dik-dik just by looking. You have to do chromosomal analysis to be able to tell. This is an old study from the 50s that shows how much worse the situation can get. This flower has been renamed, but in the older papers, it's called Clarkia deflexa. The common name for it is the farewell to spring. Okay, it's now called Clarkia bote. It's the farewell to spring. It's common in Southern California. It's an annual. Normally it outcrosses, but it can self-fertilize. And in the early 50s, a gent named Harlan Lewis collected Clarkia deflexa from 11 different populations in Southern California and tried crossbreeding them. He wasn't able to crossbreed each one of the 11 with all of the other 10. So he wasn't able to do 110 crosses, but he did as many as he could. This is where his populations of Clarkia deflexa live, by the way. That green shaded area between, let me see, up there is Monterey Bay. Down here, I think that's Big Sur. And then coming down all the way to the edge of San Diego County. That's the range. And then there's the 11 populations that he looked at. And there's what he got. That's his data plotted as a web. This is a little confusing to read, so I'm going to color code this for you. In green, you've got populations that were completely interfertile, at least through the F2 generation. He crossed them, he got fertile hybrids, he bred those fertile hybrids, and he got a second generation of fertile hybrids. So for at least 2 generations, he got fertile offspring. That is probably complete interfertility, although I guess there's a chance that you might have problems in that later generations. But at least for the first 2, we have complete interfertility. Yellow is the populations that he was able to interbreed, and he got fertile F1s. But when he crossed his F1s, the F2 either died or was degenerate or was sterile. So that's interfertility for 1 generation, but then the F2s start running into problems. The reds are cases where the F1 itself died or was sterile. And then the thin black lines, dashed means that he didn't even get any seeds. The solid line are cases where he only got a single seed. So A, as you can see, seems to be pretty reproductively isolated from all the other populations that were tested. He didn't breed AJ or AK or AB, but AC, A and D, A and E, A and F, A and G, A and H, and A and I, those are completely reproductively isolated. But you start getting into weird problems if you look at the rest of the graph. Like, look, I and D are completely fertile with each other. D and E are completely fertile with each other. But we can't close the triangle because E and I are not fertile with each other. Okay, that's kind of weird. Let's see. Where's another fun example? C and F are interfertile with each other completely. So C and F we could place in the same species. D and E are fertile with each other, and we could place those in the same species. But C and D are not fertile with each other, and E and F are not fertile with each other. Okay, so that's kind of weird. C and G are not fertile with each other. G and F are not fertile with each other, but C and F are, and so on. What I want you to get from this is the fact that there is not an obvious way that you could divide these 11 populations into completely reproductively isolated species, except possibly if you split A off from everybody else, because A doesn't seem to be able to reproduce with anybody. I mean, you could maybe make a case that CJF should be 1 species and DEI should be 1 species. But then if E and I can't interbreed, what do you do then? What do you do with a species where some populations can interbreed and others can't, but both can interbreed with a third? Okay, you see what I'm getting at here. There's no obvious way that we can split this up into perfectly isolated biological species. And again, this is the rant I kind of ended with on Friday. This is exactly what we would expect in a world where evolution is happening. There always ought to be entities that are mostly separate species, but maybe not, but mostly yeah, but eh. There ought to be a lot of cases of indeterminacy where, we honestly can't give a straightforward answer to the question, how many species do we have? That kind of fuzziness is not bad science. We live in a world where evolution's happening, and that means that we should be seeing this kind of fuzziness. The fact that it's not unambiguous what species are is actually an argument in favor of evolution happening. I think I told you about the evangelist I used to argue with in my grad school days. And he claimed that if evolution really happened, then we shouldn't be able to classify living things and everything would be a complete mess. And I said, well, yes, you know, that was, that was one of the few valid arguments that he made. But in fact, it is messy to classify species, and there are a lot of conundrums that you run into when you try to do it. And this is not a bug, it's a feature. This is what you'd expect in a living world where life has been changing for the past 4 billion years. There's some alternative concepts that have been proposed, but they're not flawless either. We talked about the biological species concept. The morphological species concept just defines a species by what it looks like, by its features. Say what you will about the biological species concept, the morphological species concept is what you use when you go out with a field guide, when you try to figure out whether that hawk that you've just seen is a red-tailed or a red-shouldered hawk. You look at the pictures in your Peterson's Field Guide to Raptors or whatever it is. And that presupposes the morphological species concept. That's what historically has been used ever since the days of Carl Linnaeus. And as we've seen, the morphological and the biological species concept don't always agree. So you can have two populations that morphologically are virtually identical, but reproductively isolated. And again, we call those cryptic species. A guy named Lee Van Valen tried to introduce the ecological species concept, where you simply define a species by the niche it inhabits. If, let's say, Western and Eastern meadowlarks are occupying the same niches in different areas, feeding on the same food, requiring the same conditions, preyed upon by the same predators, then we'll call them one species, regardless of whether they can all interbreed or not. The behavioral species concept defines a species by its SMRS. And the SMRS stands for specific mate recognition strategy. That is specifically the features that organisms use to recognize mates. An important point is, in some cases, this will overlap with the morphological species concept. If we're talking about species that attract mates with different colors or something like that. But many organisms don't live in a world where vision is the primary sense that they use. So it's very common to find populations of moths, for example, that look identical to our eyes, but they don't interbreed because they smell very differently. They use very different pheromones. And that's just because in moth world, from the point of view of the moth, their sense of smell is exquisitely well developed, and sight is not that big a deal. So the specific mate recognition strategy is not necessarily something visual. It's not necessarily something you could see. It could be calls, as in frogs, or bird songs. It could be smells. It could be dancing patterns. It could be, chemical signals between egg and sperm. But that's what you use to define species is the SMRS if you're using the behavioral species concept. There's another classic study from the 40s that shows the limitations of these alternates. A guy named Albert Blair studied the ability of different populations of different toad species to interbreed. So he collected an awful lot of toads and watched them breed and saw what he got. Traditionally, there's two major toad species in eastern North America, Bufo americanus and Bufo fowleri, the American toad and Fowler's toad. And here's a couple of examples. And I'm not showing all of his results, but one part of it is he collected Bufo fowleri in Indiana. That's a fowleri over on the left. And he mated them with male Bufo americanus from elsewhere. That's an americanus on the right. They look very similar. If you didn't know, you might consider these the same species, but are they really? Here's what he got. When he collected male fowleri from Indiana, from Bloomington, and crossed them with female americanus from Bloomington, he got everything from 95% of the eggs yielding tadpoles to only 40%. Okay. When the males came from Winona Lake, Indiana, and he crossed these with americanus females from Bloomington, none of the eggs yielded tadpoles. They're completely isolated. When he starts trucking in male toads from Tulsa and crossing his Tulsa toads, sounds like a country bar band. When he starts crossing his Tulsa toads with his Indiana toads, he gets everything from 95% fertility to only 4% of the eggs yielding tadpoles. So this is weird. So he gets everything from complete fertility between americanus and fowleri to complete sterility when he crosses americanus and fowleri. And it doesn't always seem to bear rhyme or reason. The Tulsa toads range from almost completely isolated to almost completely not isolated when he mates them with his females. Are americanus and fowleri separate species or not? Behaviorally, they probably are. The mating calls are very different. Fowleri is said to drone and americanus is said to trill. So behaviorally, you can make a case that these are separate species. Morphologically, they probably aren't. They aren't really consistent physical differences between these species. I mean, americanus is supposed to be more reddish, but I can't really see it in these pictures here. I would have no problem calling these two the same species. So maybe americanus is a little different, but the differences aren't consistent, and taxonomists have disagreed about how you should classify these toads. So morphologically, we probably have one species. Behaviorally, we have two. The ecological species concept says we have one. They overlap in their niches a great deal. Biologically, the biological species concept, you're getting everything from complete interfertility to complete intersterility, depending on where you catch on what population the toads come from, depending on where you catch them. So we have two tests that say it's one species, one that says it's two, and the biological species concept is a complete muddle. In cases like this, what taxonomists often do is define a species complex. That's basically biology speak for We have a bunch of populations that are kind of different, but not completely, but mostly, but sort of, but not, but yes, but no, but yes, but no. So we're going to call this a biological species complex and let somebody else worry about it. So we have, I suppose, the Bufo americanus species complex consisting of populations that may or may not be isolated enough to be called separate species, but disentangling them is going to be really hard. The next thing I want to talk about is called sympatric speciation, but I'm actually going to stop right there. That's a good place to take a break. I've got 9:46. We'll reconvene at 10, and I will stop the recording.